Oh, the next speaker is Jon Rask. He's going to be presenting the topic that is entitled Chemical Reactivity on In-Situ Lunar Dust for Biotoxicity Assessment. So Jon, thank you.
Thank you very much. Are you able to see my slides?
Yes.
Okay, I'll run the timer behind me.
Okay.
This activity is a collection of a lot of work by many different people. Penny and I put together some of this, but there's folks from Ames, JSC, the National Institute for Occupational Safety Health are involved. So there's many, many more people that are involved in this. And I'd like to say thank to everybody for that. So today I want to give you a flavor for, we heard about lunar dust in the context of the lunar environment and how its reactivity is important as it relates to biology. And so I will go through a handful of examples here to demonstrate that. And of course, just as a quick primer, most of what's in lunar dust is silica glass, but the material that I'm particularly interested in this talk today is the metallic iron or the nanophase iron that is relevant to chemical reactivity. And as we've heard throughout the day, the lunar environment is continuously bombarded by full spectrum UV, solar wind protons, galactic cosmic ray, solar proton events, and impact processes. And all of these things impart chemical reactivities upon the mineral grains at the surface, including the process of grinding it down into smaller and smaller particle sizes. And we have learned from other studies that transient increases in chemical reactivity and physical processes of mineral specimens have been shown to affect human pulmonary toxicity. This is in particularly in reference to silica. So we've done a lot of studies with silica, and in the studies that I'm going to be talking about in the future, we actually worked with Apollo 14 specimens. So what do we mean by chemical reactivity? Simply put, it refers to radicals that exist on mineral grains that can interact with oxygen and water. And these are referred to as reactive oxygen species, including hydroxyl radical, superoxide, and peroxide. And we'll hear more about that later in Donald Hendrick's talk. And biological exposure to mineral grains and its chemical reactivity will elicit an inflammatory response that leads to the production of reactive oxygen species in the cell itself. And so we have explored ways to bake material or expose it to water and oxygen to decrease its chemical reactivity. And of course, the other phenomenon in the lunar environment enhances it, as mentioned before. So are there ways that we can study this with samples we have? Yes, we have samples from the Apollo missions, and of course, there's samples from Russian missions. And of course, not pictured here is the samples that have been brought by the China mission. And we have learned over the last 30, 40 years that lunar dust is definitely chemically reactive. And Apollo specimens have been used for a variety of studies, looking at infectivity and toxicity studies. And some of the earliest ones looked at dissolution changes and UV optical properties that were altered upon analysis. But the most recent and more dynamic results in terms of crushing lunar dust was done by Billy Wallace and others. They discovered that it's much more reactive in its freshly crushed state, similar to silica. And one of the most profound visual references, in my opinion, is the fact that we have learned that hematite has been observed on the moon. And we also know that hematite nanoparticles are toxic to bacteria. So just to further emphasize this, in the north and south pole regions of the moon, the colored yellow to red areas are enriched in hematite. And so this is another clue that there is a reaction going on, or in the past, that is combined iron oxides and nanophase irons are reacting with oxygen and water to form hematite. So we know that there's chemical reactivity going on in the moon's environment. And much of that is attributed, as we've heard throughout the day today, about nanophase iron, and that is something that's appearing in the rims of each of these little tiny mineral grains. And up in the upper right corner, to emphasize the fact that there is elemental iron in there. Larry Taylor used to always carry a magnet around to demonstrate how each individual particle of lunar dust is attracted to a magnet, demonstrating the fact that there's elemental iron present. And another thing that came out of Wallace's study and a lot of work that Larry Taylor and others did was the point of reactivity relates to the mineral maturity, that is how much elemental iron is present in the samples. And hydroxyl radical scavenging assays have shown that mature samples are much more reactive than immature samples. And it's important to note that lunar soils at the surface become more mature over time due to space weathering. And we know that the lunar surface is very different all over the moon. And so the site of location where you're working is important in terms of analyzing that actual sample. So there's been a lot of work done on reactivity analysis of lunar samples and simulants. And the main takeaway here is that I want to emphasize that both NASA and Russia have independently observed that Mare samples contain more nanophase iron than Highland samples. And as we mentioned before, Mare samples appear to be more reactive than Highland samples. And also, there's been a number of biotoxicity studies that have been performed both by the US and Russia. And Mare samples have been reported to be more biotoxic than Highland samples, although the Russian studies were a little bit flawed because some of their particle size controls were less effective than the LADTAG studies, which were highly particle size controlled. And so our take-home message here is that lunar dust chemical reactivity is predicted to be enhanced in the lunar environment. And to drive this home, we've done some biological studies in rats. Biological response is going to increase as a function of dose, and the dark line on the right is considered a passivated or low reactivity sample. And if we chemically activate lunar material, it will shift the biological dose response curve to the left in what we would call a chemically activated dose response curve. And this is in fact what we observed in a series of animal studies in rats, where we injected small doses of lunar material from the Apollo 14 field site into animals. And we used 3 different preparations of lunar material: ball milled, unground, and jet milled, 3 different preparations of the same material. It was particle size controlled. Roughly about 2.5 microns or so was injected into the lungs of these rats. And we measured the inflammatory response of the cells that responded to this injection of dust in the lungs of the rats. And the short story is that that material, which was more highly chemically reactive as assayed by the hydroxyl radical scavenging assays we've used, was more biologically toxic. Although the difference was small, it was highly statistically significant in terms of it being more impactful in the lungs of the rats. So this is a very important observation that we had made here, and it further emphasizes the importance of chemical reactivity as it relates to its biological effects, in this case in the lungs of rats. But more broadly speaking, chemical reactivity is critical, as we've heard also in this session, to human health, but also space biology and astrobiology. And it's important to remember that radicals contribute to cell death. They contribute to signal transduction disruption, gene expression changes, and oxidative damage to nucleic acids, proteins, and lipids. And this is important in the whole scope of all biology. And we also want to point out that radicals rapidly react with organic molecules, and they interfere with assays and other methods. And in some cases, they can damage instrumentation. And it's important also for us to be considering that Earth-based analogs, simulants, and archived lunar dust do not fully represent the picture of in situ reactivity and biological effects. What we really need to do is take a measurement on the lunar surface and compare that to what we can measure here on Earth. And even more broadly speaking, in terms of engineering and hardware development, future engineering tests that use hardware destined for the moon that is sensitive or is going to be involved in some aspect of chemistry of lunar materials, those tests of that hardware must be conducted on appropriately treated specimens, that is, those that most appropriately represent material from the in situ lunar environment. And with that, I'll stop and thank everybody for your attention.
Thank you, John. Interesting topic and talk